You remember there is a cytosol, a solution inside of the cell. So that cytosol has 9 grams of salt in 1 liter of that solution, or it will be concentration 0.9%. So that solution is called isotonic. Any solution that has the same amount of salt like it is inside of the cell, it will be called isotonic. So any isotonic solution has 9 grams of salt per 1 liter of the solution, which is equal to the concentration of salt inside of the living cell. Some solutions can be hypertonic. It's a solution that has more salt, more than 0.9%. And finally, there are hypotonic solutions having less salt than 0.9% or zero salt at all, like pure water. What are you gonna do now? That red circle, imagine it's a red blood cell. We take 3 red blood cells and place them in 3 different solutions. This red blood cell, inside of it there is an isotonic solution, 0.9. So this red blood cell was placed in a hypertonic solution. Concentration of salt outside of it is much higher. Maybe it's 1%, maybe 2, 3, 5% of salt. What happens with the cell in that solution? Where water will be moving? Think about it. Where concentration of water is higher? It's high inside of the cell. So water will be moving out of that red blood cell, and red blood cell will shrink and finally die. The second red blood cell was placed in a hypotonic solution, where outside there is a solution with a very low concentration of salt. What happens with that cell? Water will be moving into the cell from high concentration of water to low concentration of water. The cell will swell and finally burst and die. And next one, this red blood cell was placed in an isotonic solution. Small amount of water will be moving here and there, but generally there will be not a lot of movement of water. That cell is just fine and happy. Okay, here is a picture from your book that illustrates what happens with red blood cells in hypertonic, isotonic, and hypotonic solutions. Please know that and be able to explain. Okay, here is a summary of your passive process presented as a table this time. So simple diffusion, no ATP used, movement of oxygen, for example, oxygen through phospholipid bilayer. Phospholipid diffusion, example would be movement of glucose into cells. Facilitated diffusion, its movement through proteins. Again, no ATP used. And finally, osmosis, no ATP used, but that would be a movement of water. Now we will talk about active processes. So there are 2 types of active processes or movement of substances across the cell membrane using ATP energy. There is active transport itself and vesicular transport.